Hello friends, hope all are fine. Welcome to my channel Sam Biology Plus. Today we will discuss about the principle and applications of phase contrast microscope. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. A phase contrast microscope is a specialized light microscope that enhances the contrast of transparent, unstained specimens such as living cells and microorganisms by converting phase-shifting light passing through them into detectable amplitude change. Amplitude determines the brightness and intensity of light. So when amplitude increases, intensity and brightness also increases. Phase contrast microscope was invented by Fritz Zernike. The technique of phase contrast is widely applied in biological and medical research throughout the fields of cytology and histology. As such, it is used to visualize living cells, tissues, and microorganisms. In addition, it is used to examine internal cellular components such as membrane, nucleus, mitochondria, spindles, mitotic apparatus, and chromosomes. This methodology is also employed to diagnose tumor cells, growth, and to study the behavior and dynamics of cells and tissues from both plants and animals in culture. Key components of a phase contrast microscope are an annular phase condenser and a phase contrast objective. The annular phase condenser, which produces a hollow cone of light, is composed of the substage condenser and an annular diaphragm. This diaphragm is located at the front focal plane of the condenser lens. The annular diaphragm, which is also known as condenser annulus or phase annulus, is an opaque, flat, black, and light-absorbing plate containing a transparent annular ring. Transmitted light from a specific source only passes through the transparent ring. Light do not passes through the remaining area, which is opaque. The other component is the phase contrast objective, which consists of a specialized phase plate and typical lenses for magnification. The function of the annular phase condenser is to transmit a hollow cone of light which strikes the specimen. Transmitted light from the source passes only through the transparent ring and then passes through the condenser lens and is then focused on the specimen as a hollow cone of light. Because the thickness and the refractive index of the specimen is higher than its surrounding, the light passing through the specimen is retarded by approximately 90 degrees. Retarded means the waves slow down. As a result, its speed, amplitude, and brightness decreases. So the light passing through the specimen is refracted in different directions depending on the optical density and the refractive index of different structures that are present within the specimen. The remaining light, which is linear, passes through the background or between cells. This light constitutes the background or surrounding light. So here we have diffracted light which comes from the specimen and the background or surrounding light. The diffracted light and the surrounding light then passes through the objective and then through the phase plate. The phase plate is a specialized structure which produces phase contrast, either positive or negative. There are two types of phase plates. Number one, positive or dark contrast phase plate, which has a thinner circular ring that selectively advances the surrounding light by 90 degrees. Such type of phase plate produces positive phase contrast in which the specimen appears dark against a bright background. Number two, negative or bright contrast phase plate which has a thicker conjugate area that retards the phase of surrounding light by 90 degrees. Negative phase plate produces negative phase contrast in which the specimen appears bright against a dark background. In phase contrast microscope, the diaphragm is matched in diameter and optically conjugate to the phase plate. The plate contains a specialized ring known as phase ring, which contain materials that is responsible for shifting the phase of light. The area of the ring is known as conjugate area, whereas the remaining region is known as complementary area. The diaphragm and the phase plate have been positioned in such a way that only the direct surrounding light will pass through the phase ring, whereas the diffracted light that is coming out of the specimen will pass through the complementary area. The phase of a sinusoid wave is the angular displacement that specifies its position relative to a reference wave. A wave can be retarded or can be advanced. When this happens, we say that there is a phase shift. When the wave is advanced, then the phase shift is positive. But when it is retarded, we say that the phase shift is negative. The wave which undergoes a positive shift is known as the leading wave. On the other hand, the wave which exhibits negative phase shift is the lagging wave. The phase shift is expressed in terms of angular displacement, which is calculated by determining the angular difference between corresponding points on two waves. It is measured in degrees or radians. One complete cycle of a wave corresponds to 360 degree or 2 pi radians. This figure illustrates what is phase and phase shift. In this graph, this represents one full cycle of a wave. At this point, the angular displacement is 90 degree or pi by 2. At this point, it is 180 degree or pi. But at this point, the angular displacement is even higher. It is equal to 270 degree or 3 by 2. And one full cycle represents 360 degree or 2 pi radians. This figure illustrates positive and negative phase shift. In this graph, the black wave is the reference wave and the blue wave is the one which has undergone positive phase shift. So it is known as leading wave. On the other hand, the red wave, which has been shifted to the left, has undergone negative phase shift by 90 degree. So red wave is the lagging wave. So in a phase contrast microscope, depending upon the nature of the ring, the surrounding wave can either be advanced or retarded. When retarded, negative phase contrast develop, but when advanced, positive phase contrast develop. As already said that when light passes through specimen, it is retarded approximately by 90 degrees. That means the waves coming out of the specimen have undergone a negative phase shift. These rays are diffracted in different directions depending on the optical density and refractive index of different structures present within the sample. In positive phase contrast microscope, the phase ring is thinner. As a result, direct or surrounding light passes through the phase ring is selectively advanced by that means the direct or surrounding waves have undergone a positive phase shift of 90 degree. On the other hand, the diffracted waves have undergone a negative phase shift of 90 degree. Now the diffracted waves and the surrounding waves are out of phase. In this case, destructive interference takes place between the diffracted wave and the direct and surrounding waves at the image plane. As a result, the specimen appears darker against a bright background. By contrast, in case of negative phase contrast microscope, the phase ring is thicker. As a result, direct light passing through the ring is retarded by 90 degrees. It is because both diffracted light and the direct light have undergone a negative phase shift. Now they are in phase. As a result, constructive interference takes place between two types of lights. As a result, a resultant wave is produced which has a greater amplitude. This figure illustrates constructive interference. These are the two waves X and Y, and these upper points are known as crest. The upper points are known as crest and the lower point is known as trough. This height is known as the amplitude. You know that amplitude determines intensity and brightness, and the distance between two adjacent crests or between two adjacent troughs is known as wavelength, which is designated by the symbol lambda. So these are two waves which are completely in phase. In phase means the crest of one wave meets the crest of the other wave. So because these two waves superpose, the interference which takes place between them is constructive. As a result, the resultant wave which develops has a greater amplitude which increases the brightness and intensity. So whenever constructive interference takes place between diffracted light and direct or surrounding light, negative phase contrast develops. As a result, the specimen appears bright on a darker background. Again, to inform that when both the diffracted light and the direct light have undergone negative phase shift, constructive interference takes place between them at the image plane. In this case, the waves are in the same phase. In case of constructive interference, two waves add up and gives rise to a resultant wave which has a higher amplitude or brightness compared to the corresponding waves. This gives rise to negative phase contrast in which the specimen appears bright but the background appears dark or dim. On the other hand, when diffracted light undergo negative but the direct light undergo positive phase shift, destructive interference takes place. This is because the waves are out of phase and in this scenario they cancel, that gives rise to darkness. This is known as positive phase contrast in which the specimen appears dark on a bright background. So phase contrast microscope enhances the contrast of transparent and unstained specimen by converting phase shift in light passing through them, which is not detectable by human eyes, into brightness or amplitude change, which is well detected. So friends, this is all about the principle and applications of phase contrast microscope. For more updates, subscribe my YouTube channel, Sam Biology Class.